Thank you, Dr. Bullion. Can you hear me?
Yes, indeed. We can hear you very well.
Okay. So my camera is not, it's kind of blurred, so I will keep it off. So yeah, good day, everyone. So welcome all to your this last presentation of this session. I'm really excited to share these interesting results of my PhD research here. This presentation, I titled it as Safeguarding of Broiler Chickens from Lethal Bacterial Infections using CpG-ODN to Initiate the Trained Immunity. So, as we all know that bacterial diseases cause significant losses to the industry. So this is my presentation outline. It will have two objectives. One objective included methodology and results. So here is the health and economic impact of bacterial diseases. So here actually we are focusing E. coli and Clostridium perfringens. And also I want to mention here, so in this presentation, we are not focusing on one particular disease. So all the bacterial diseases we are concerning, but we challenge with E. coli and Clostridium perfringens here, so that's why I am emphasizing it here. So these bacterial infections increase the mortality and clinical signs and cause the significant losses to the industry. And also it increased the production cost. And not only that, it increased the serious risk to the human health through the foodborne illnesses. So the existing preventative measures like antibiotics, so there's a limitation of usage those preventative measures because of rising of antimicrobial resistance. And also chicken farms of Canada recently eliminated of preventative usage of antibiotics. Therefore, industry is urgently looking for alternative strategies. So speaking of alternatives, I want to highlight about the CpG-ODN here. So what is the CpG-ODN? It name defines cytosine phosphodiester guanine oligodeoxynucleotide. And this is a synthetic homologous to bacterial DNA. And because of that, this acts as a bacterial DNA in the immune system, but without producing any diseases, it can stimulate the immune system to produce different cytokines and protect especially the vertebrate animals against different diseases. And this is a non-methylated, short, single-stranded synthetic DNA molecule. How CpG-ODN mediates its cell signaling? The CpG acts as a pathogen-associated molecular pattern, and it is recognized by pattern recognition receptors like TLR21 in chickens. So upon this binding, it stimulates number of intracellular signaling cascaders like NF-kappa beta pathway. And at the end, it gives signals to the nucleus to produce more pro-inflammatory and inflammatory cytokines, which initiate the protective immunity in chickens. So there are a number of proofs of immunoprotection mediated by CpG-ODN chicken in our lab. And also previously a study have shown that it can shift the serum metabolic profiles in chicken following CpG-ODN administration.
Do you know how to reduce the voice?
Here we go. Are we back?
Why is the voice is not going?
Hello.
Hello. Something happened with our Zoom connection. I'm not sure quite what happened. You were on.
Oh.
You were on. I'm so sorry. it just without all of a sudden everything went quiet, and then my Zoom is trying to connect. So my apologies, everyone. It looks like we're back online.
Okay.
You were.
So I'm still sharing the screen.
You are sharing your screen now. I apologize to everyone, but I don't know quite what happened there. That was a couple of moments of kind of freaking out. So if you'd like to
which slide now I am what do you want
if you go back.
Sorry, Brenda, from my side, we didn't get a very long interruption, sorry.
Oh, you didn't? It was completely gone for me. So I don't know if that means it was I will I don't know what to tell you.
Well, I guess that Iresha can probably go back one slide just to make sure that there was no lost in translation, if I can say that.
I think that's fair. I apologize. It was the system went offline completely for me. So
It's okay. It's a busy morning.
yeah, it has been. It has been. So I will off you go. I will stop interrupting.
So
Thank you.
So which slide I should start somewhere here or?
Yes, well, if you can go with the proofs and then from there.
This here?
Yes.
Yeah. Yeah, actually, I was mentioning that we have in our lab, we have done a number of studies to prove that immunoprotection mediated by CpG-ODN chicken. So, and in this study, this particular study, we have focused on whether the CpG can initiate the trained immunity. So let me ask you, if you all are familiar with this innate immune memory, because we all know that adaptive memories exist and which is mediated by antibodies, and it is a specific memory. But this is quite fascinating about the innate immune memory because when the primary challenge, or it can be a vaccine or infection, so this immune system gets stimulated, then the innate immune cells undergo training period by metabolic rewiring and the trained epigenetic reprogramming. So these trained immune cells, once they're exposed to the secondary exposure, like unrelated pathogens, or it can be any pathogen, it shouldn't be the specific pathogen, same as the primary exposure. So these trained immune cells have ability to induce the trained immune response and then protect against that unrelated pathogen. This is a non-specific and long-term protection and against non-related pathogens. And also, I want to give you a very interesting example here. You know that when during the COVID pandemic, there were some countries which had high disease severity and mortalities, but whereas the other countries has less diseases and the less mortality rates. And the scientists have looked into that, and then they found that these people which had low mortality rates have exposed to the BCG vaccination at their early stages of life. And this is because of the innate immune memory. So this is quite fascinating, and then we wanted to see whether chickens also have this kind of amazing ability. So here's the objective. So we wanted to discover an effective treatment method by empowering the innate immune cells to control economically important bacterial diseases in broiler chickens, because this is a non-specific memory in the chickens. And then we have two objectives. First, to administration of the CpG-ODN twice to induce trained immunity through the metabolic reprogramming against E. coli infection. So I'm moving to the first objective. So we injected 2 CpG doses, and then we wanted to check by the end of their production cycle if these broilers gain the protective immunity. So this is what we did. Day 1 and day 4, birds were injected CpG-ODN intramuscular route. And then after day 1, day 7, and day 21, the blood samples were collected from the same set of birds. And then we quantified the mitochondrial oxidative phosphorylation and the glycolytic rate using seahorse XFP analyzer. This method is also we optimized in our lab, and it's a real-time quantitative method. And then at the end of the life cycle, or the mostly end of the cycle of the broiler chickens, day 27 of age, birds were challenged with E. coli. And also we separated the immune cells using max separation method, separated monocyte macrophages, T cells and B cells, also quantified the mitochondrial OXPHOS. So in the results, it was very interesting. We have seen the mitochondrial oxidative phosphorylation in the CpG-ODN group had significantly higher in all the time points up to day 21. But in the cellular glycolysis, we have this pattern. The CpG group had significantly higher at the first 2 time points, but at the end of the time point, it's decreased. So there were no significant difference between groups. So this means the immune cells undergo metabolic reprogramming, which shift the immune cell metabolism from cellular glycolysis to the mitochondrial respiration. And this is another interesting finding, because at the end of the experiment at day 27, we challenged them with the E. coli, and the survivability you can see here in these slides, a cumulative clinical score significantly lower in the CpG group, and also significantly higher survival rate in the CpG group compared to the saline birds. And these are the flow cytometry results. We purified the immune cells, monocyte, B lymphocytes, and T lymphocytes in the both groups. And here shows the metabolic outputs of individual cell types of mitochondrial oxphos. And this line shows that this is the basal respiration. Here's the maximal respiration. And this, you can see the monocytes have higher, significantly higher OCA activity or mitochondrial respiration in the CpG group than in the saline group. B lymphocyte, we have seen it is increased, and the T lymphocyte, we couldn't see much difference. So it means monocyte is kind of playing a major role behind this activity. And now I'm moving to the second objective. We delivered in novo CpG-ODN prior to the CP vaccination. This is the Clostridium perfringens live vaccination to instigate the trained immunity against necrotic enteritis. So we wanted to see if it affects the vaccinated birds. So this is what we did. At the day 18 of incubation, birds were injected in novo CpG-ODN in the embryos. And then after that, day 1 and day 10, birds were administered the vaccination, live CP vaccination, and then blood samples were collected at day 3, 14, and 21 after the booster. Then immune cells were separated to quantify the mitochondrial oxphos and cellular glycolysis. So what we see here at day 3 and then day 14, day 21 post-booster, the pink line shows the CpG plus vaccinated group, significantly higher in all the time points up to day 21 post-booster. This mitochondrial activity is significantly higher than the vaccination alone and the CpG alone group. In the cellular glycolysis, this is quite interesting. At the first time point, it was significantly higher, but in the other two time points, we couldn't see much difference here. So metabolic reprogramming happens in these cells. And also in the survival data, we observed that significantly higher protection in the CpG plus vaccinated group compared to the vaccine alone group. So in the conclusion, what we can conclude from our results here, in the first objective, we have seen only 2 administration of the CpG-ODN administration at early ages, like day 1 and day 4, induced the trained immunity in these chickens. And this is followed by the metabolic reprogramming, shifted the immune cell metabolism into the mitochondrial respiration pathway from the cellular glycolysis in the long-term aspect. And this instigated trained immunity was able to protect broiler chicken at the end of their life cycle at day 27 against E. coli infection. And this is mostly mediated by monocytes. And the second objective, we can conclude in novo delivery of CpG-ODN induced the trained immunity by metabolic reprogramming in vaccinated broiler chickens to protect against necrotic enteritis or against Clostridium perfringens. So this is the take-home message graphical summary for you. So we have done 2 experiments, CpG-ODN, 2 injections here and the novo injection here. And these both scenarios can train the innate immune cells via metabolic reprogramming, glycolysis, and the mitochondrial respiration. And then at the end, they allows chickens to gain protective ability against the deadly bacteria, especially E. coli and Clostridium perfringens. So what are the benefits? Obviously, it's decrease the disease incidence, increase the production, decrease the production cost, and also it protects us from the foodborne illnesses. So this initiated non-specific trained immunity, we can use this against other economically important bacterial infections. So I would like to acknowledge these people, my supervisor, Dr. Susanta Gomes, and also our Gomes Lab Research Group. Thank you, everyone, Animal Care Unit, users, graduate scholarship, and our funding sources. Thank you so much, everyone, for attention, and I'm happy to answer if you have any questions.